as I have found with most plants that this is necessary in order to grow the strongest and healthiest specimens. Other factors, including nutrition and moisture, are of course also very important, as well as air, and especially humidity when it comes to growing orchids. Humidity, though, is also a problem in keeping the film in the cameras for the long periods of time necessary to photograph a growing plant. So the long plastic hoses bring conditioned air to the aluminum hoods over each camera. Time-lapse photography is just the opposite from slow-motion pictures. And here you can see a bird of paradise burst into bloom in a matter of a few seconds. In taking time-lapse pictures of plants growing, I've encountered some interesting exceptions to the general rules of nature. Photographing the growth of a banana was a very good example. Not having any bees in the greenhouse, I thought I might have to artificially pollinate the banana blossom. So I got out my little camel's hair brush, but I couldn't find any pollen. I contacted the research people at the United Fruit Company and learned all about a banana being what's called a parthenocarpic fruit. This means it produces mature fruit without the blossoms being pollinated. The banana is one of the oldest fruits to grow on this earth, and it is thought that originally it did produce pollen, but it no longer does. It is male sterile, and this is a great handicap to the banana growers because it means they cannot cross-pollinate or hybridize bananas to develop new, improved varieties. To take this picture from the emergence of the first shoot to the mature fruit required 10 cameras operating continuously day and night for 2 years. Here you see some of the additional attachments and gadgets that I borrowed from the works from the old kitchen clock that I used to make my first automatic timer. Before building the ultraviolet transmitting greenhouse, I made my time-lapse pictures in this basement studio that I called my ivory cellar. The cameras and subjects were under this skylight, which made it necessary to supplement the restricted daylight with various types of artificial light sources. Each time a single frame is exposed in any of the cameras, the supplemental growing lights are turned off momentarily, the overhead shutters closed to shut out the daylight, so I have the same amount of photographic light for an even exposure day and night and regardless of the weather. Here is a time-lapse picture speeding up the action of taking time-lapse pictures so that you can see how the cameras move forwards and backwards, they tilt up and down and pan from side to side as programmed in advance to follow the growing subjects. At normal speed, the hand cranks turn about the same speed as the hand on a clock, so this will give you an idea of approximately how much some of the action is speeded up through time-lapse photography. Here is a time-lapse picture of an iris, which is a nocturnal or night-blooming flower. This is a hibiscus, which blooms during the daytime. And here is a camellia that blooms according to a relationship between temperature and the dark nighttime period. The nights must be very cool to bring the camellia into bloom, but the temperature during the daytime is relatively unimportant. It is just the opposite, though, with the azalea, which commercial florists can force into bloom ahead of the normal season by controlling the temperature in relation to the light period. Here is a cactus that grows in the hot, arid desert. Plants, as well as many animals, have adapted to various environmental conditions. In contrast to the cactus, here is a water lily. This is the passion flower. And here is an orchid that requires a warm, moist atmosphere. These flowers all grew quite well without any difficulty. But the first problem that I encountered with any possible scientific significance came as the result of attempting to grow a pumpkin from the emergence of the first shoot to the mature pumpkin for Walt Disney's film Secrets of Life. I planted some pumpkin seeds in large boxes of soil and placed them under the skylight in the ivory cellar where the young plants received some direct sunlight around noon when the sun was directly overhead, but not during the morning or afternoon as they would outdoors. So I installed some ordinary fluorescent light fixtures with cool white fluorescent tubes that are very rich in the yellow-orange part of the spectrum because of more energy in these particular wavelengths. They're designed this way to give a warmer tone to cosmetics and interior decorations. Here you see the tendrils reaching out for some solid object for support. As soon as they find something, they wrap themselves around and get a good firm grip, then start winding themselves up to form a natural spring that snubs the plant down, and they won't break so easily. The first thing that I learned about a pumpkin was that it is a monoecious type of plant, meaning that it produces the staminate and pistillate blossoms separately on the same vine. Here you see some of the staminate blossoms, extra large, healthy specimens. The leaves are nice and green right to the very tip point of each leaf, indicating no apparent nutritional deficiencies. But while all the staminate blossoms grew so nicely, I suddenly realized that all of the pistillate blossoms, with their little embryo of the pumpkin right under the flower, would only reach this early stage of development, and then stop right there and dry up, turn black, and drop off the vine. So I didn't get any pumpkins. The second year, my lights were old and beginning to flicker. So without asking for one type of light or another, I bought some new fluorescent tubes. And the second year, all the pistillate blossoms grew very nicely, and all the staminate blossoms dried up and dropped off. This was just the opposite from the previous year. I repeated this experiment a number of times and found that I could obtain 100% staminate or pistillate blossoms on a pumpkin vine by simply supplementing the restricted daylight with either cool white or daylight white fluorescent, which I happened to be using the second year. Daylight white fluorescent is strong in the blue end of the spectrum. Chinchilla breeders are now able to obtain up to 85 or 90% male or females in the litters, depending on the lights used in the breeding rooms. Here at last is the pumpkin that was in Walt Disney's film, Secrets of Life. And here are the two types of light, side by side, next to the skylight. The large seed company asked me to make some time-lapse pictures of morning glories for one of their films, and I thought this would be a very simple project and promised the pictures in about 2 weeks. But well into the second year, they were becoming a little irritated at all the excuses I was offering as to why I could not deliver the films. The reason was the buds would reach the stage where I would expect them to open by the following morning, but instead they would simply shrivel up and collapse. This was the first commercial project undertaken in the new plastic greenhouse, so I tried it again in the old glass greenhouse, but had the same results. Then on one of my gardening television programs, I was interviewing a commercial florist who specialized in bringing chrysanthemums into bloom the year round by controlling the periodicity of the light, and also timing the blooming of poinsettias for the Christmas trade by interrupting the night dark time period with artificial light. Meanwhile, I discovered that the morning glories are a night blooming flower, so I decided to hang a light out on the garden fence where they'd been blooming perfectly normally all summer. I connected it to the automatic timer in the greenhouse so it turned on for a few seconds every 5 minutes during the dark nighttime period. The next morning, within a perfect circle around the light, the buds were collapsing as they had in the greenhouse. Then I happened to run out of the regular type of film that I was using, and the only film I had available was daylight type Kodachrome. That meant changing the photographic lights to the slightly bluish ones to match the daylight film. The buds began opening just a little, which was the first encouragement I had had in almost 2 years. The only difference that I could see was in the light. So I decided to put some additional blue filters over the slightly bluish lights, and of course it made the picture very blue, but it also filtered out the red, or the longer wavelengths from the spectrum of the photographic lights, interrupting the normal dark time period. By filtering out this part of the spectrum, the buds then opened perfectly normally. But the pictures were so blue, I tried placing a red filter over the camera lens to correct the color. To begin with, I had too strong a red filter, and it made the flowers look purple. So by cutting down on the strength of the red filter over the camera lens and keeping the blue filter over the lights, I was finally able to obtain a reasonable color balance photographically, but still basically filter out the red or longer wavelengths from the spectrum of the photographic lights interrupting the normal dark nighttime period. and here at last was a picture of the morning glory. This indicated that this biological response is not to the total spectrum of light interrupting the dark period, but rather a narrow band of the longer wavelengths in the red end of the spectrum. I was asked to make some time-lapse pictures of tomatoes growing for some of the old-time tomato growers in the northern Ohio area. I learned that their tomato plants seemed to be more subject to tomato virus that you see affecting this plant during and following long periods of cloudy weather in the wintertime and in their glass greenhouses. Ordinarily, this virus spreads so rapidly that on the first signs of it, they rogue the plants out and burn them. They happen to have several plants growing in their experimental greenhouse just beginning to show this virus condition, and they were very happy to have me take them home with me from their glass greenhouse to my ultraviolet transmitting plastic greenhouse that lets through a more complete spectrum of the full natural sunlight. I continued to use the same fertilizer program that they were using, and not only the plant being photographed, but all 6 of the plants I brought back perked right up, started putting forth healthy, vigorous growth. They set buds and produced tomatoes, which was considered unheard of and impossible according to the old-time tomato growers. The process of photosynthesis is sometimes described as being a conversion of light energy into chemical energy, and viruses are often referred to as being an abnormal chemical or an abnormal chemical compound within the cell. Accordingly, I believe that these pictures and others that I have taken strongly suggest the possible relationship between the abnormal chemistry associated with viruses responding through this process of photosynthesis to an incomplete or unbalanced light energy source, and that there may be a very direct relationship between viruses and light energy that should be further studied. Here you see the tomato developing, and as you watch, you will see it turn a nice red color as it ripens. It is interesting how different species of plants respond to different light conditions. A tomato can be picked green from the vine and placed on a shelf in a dark closet, and it will develop a red color. Whereas an apple left growing on the tree would not develop a red color until a glass skylight was removed and replaced with ultraviolet transmitting plastic, indicating that the ripening of an apple is dependent on the ultraviolet wavelengths that do not penetrate ordinary window glass. Here you see the streaming of the chloroplasts within the cells of elodea grass. I have found that under full natural sunlight, all of these little chloroplasts get into a streaming pattern and go in an orderly fashion around and around to each end of the cell. But if the light is filtered through ordinary glass that cuts out the ultraviolet, or, as in this case, an ordinary incandescent microscope light source lacking the ultraviolet was used, many of the chloroplasts drop out of the streaming pattern and form a sluggish clump in one part of the cell or another. When I placed a red filter in the light source, restricting the wavelengths to just the longer ones that we see as red, some of the chloroplasts responded in their normal pattern, some dropped out of the streaming altogether, and others started shortcutting across without going all the way to the end of the cell. This would appear to be affecting the normal process of photosynthesis and the resulting cell chemistry. I then decided to change the red filter and insert a blue one, letting through the shorter wavelengths. And as you can see, some of the chloroplasts continue to respond normally, some have dropped out altogether, but those shortcutting go down to the upper corner before they make their shortcut. I removed the color filters and added a very low level of long wavelength ultraviolet or black light to the ordinary incandescent microscope light source to come as close as possible, in a crude way, to the full natural spectrum of sunlight. And you see just about all of the chloroplasts resuming their normal streaming pattern. At the end of the day, no matter how much I would increase the light intensity, the chloroplasts would just run down like a dead battery and refuse to respond any further until they had had their normal dark night rest period. This points up the importance of the seasonal changes in the length of day and darkness, or what is commonly referred to as the periodicity of light. Chrysanthemums normally bloom in the fall of the year as the length of daylight gradually shortens and the dark nighttime period increases. Many florists take advantage of this biological phenomenon and force their chrysanthemums into bloom ahead of the normal season by artificially shortening the long daylight hours They cover their plants with black cloth about 4:30 in the afternoon and keep them covered for several hours after sunrise the following morning. Blooming can be delayed by turning lights on and artificially lengthening the short daylight periods of fall and winter. This is how florists control the blooming of chrysanthemums so that they are available every month of the year. The blooming of poinsettias is also controlled by light, so they will reach their peak of bloom just in time for Christmas. In the early 1920s, a Canadian zoologist by the name of William Rowan discovered that the migration of birds is also controlled by the seasonal changes of the length of the day and night periods. The poultry industry has learned that egg production can be increased by lengthening the daytime periods with artificial light, and especially during the short daylight hours of the winter. More recent research by a number of scientists has indicated that light entering the eyes influences the pituitary and pineal glands by means of neurochemical channels that are independent of the optic nerve. These master glands control the entire endocrine system and the resulting basic body chemistry through the production and release of hormones. Thus, it appears that the conversion of light energy into chemical energy through the process of photosynthesis in plants carries on into animal life in a much more improved and more sophisticated way. Here is a pigment epithelial cell from the retina of a rabbit's eye, as seen through a microscope. These cells are located right behind the rods and cones, but are thought to have no visibility function. These pictures were made in connection with a drug toxicity test to see what the effects of various tranquilizing drugs might be that were known to cause various side effects. I very quickly found that the color of the filter used in the light source of the microscope to increase the contrast of the pictures photographically had a far greater effect on the cells than the drugs being tested. With the blue filter, the cells seem to go through all sorts of contortions, as you see here. With the red filter, the response was entirely different, an apparent weakening in the cell walls or the cell membrane, which would rupture, allowing the cytoplasm or the contents to run out and, of course, killing the cells. I used a water cooling condenser and a heat-absorbing filter, so I'm certain it was not a matter of a difference of temperature. Whenever a color filter was used more than a few hours, there would be no more normal cell division or mitosis. This raises a question of what effect colored filters like sunglasses may have when placed in front of the eyes. Even without a color filter, but using an ordinary incandescent light source, in a matter of a few days, the pigment granules would become sluggish and there would be no further mitosis or cell division. The punch mark in the film indicates I've added a small amount of long wavelength ultraviolet light, the same as I did with the chloroplasts. And then the sluggishness of the pigment granules would be broken up and the cells would continue their normal cell division process. Here again you see the cells in their normal state, and the second punch mark indicates a higher intensity of ultraviolet, and immediately you begin to notice a very abnormal response and finally a rupturing of the cell membrane. This is from too much ultraviolet. These same cells appear to be more active in the morning and gradually slow down toward evening. They too must have a dark period just like the chloroplasts in the cells of a leaf. Then the following morning they are more responsive to light energy. Here are heart cells from a chick embryo, and again with a blue filter you notice a complete change in their appearance and metabolic rate of activity. After seeing these pictures, several well-known virologists have commented that this reaction resembles very closely cells being attacked by viruses. They could hardly believe me when I explained that I could consistently repeat these responses by simply placing a blue filter in the light source of the microscope. This is another possible indication of a relationship between viruses and light energy. With a red filter, again an apparent rupturing of the cell membrane, allowing the cytoplasm to run out and killing the cells. These pictures show similar growth responses in animal cells to different wavelengths or colors of light that might otherwise be more closely associated only with plant cells.